we would be starting in plant biotechnology. We would be starting with relation to maybe some of the sort of processes in recombinant DNA technology. Now, in the among the processes of recombinant DNA technology, the topic of the time is selection of transformed cells. We already know that we had the sort of a foreign gene of interest, foreign DNA of interest. We had used restriction enzymes, we had used cloning vectors, and later on we knew how to cut the sort of a gene of interest and load it into a sort of a vector. And a few more things which you have already heard is about the competent host. So, after the gene gets transformed, after the gene, the recombinant gene gets into the bacterial cell, then the bacterial cell is termed as being genetically transformed. So, after we transform it, we need to have a sort of a competent host for delivery of your sort of a foreign gene of interest. Now, in the selection, we have to exactly know what is genetic transformation, how are cells selected. Now, if you recollect, whenever we have a recombinant DNA, or a foreign DNA or an alien DNA, otherwise it is also called the gene of interest. We have a recombinant DNA which has already undergone some sort of a change. Now, normally this recombinant DNA will combine with an ampicillin resistant gene. If you recollect, we had an ampicillin resistant gene and a tetracycline gene in our PBR322. So, this can combine with an ampicillin resistant gene. So, this ampicillin resistant gene is termed as a sort of a selective marker. It is termed as a selective marker. And it helps, the gene, ampicillin gene is a selective marker. What does it do? It helps in selecting a transformed cell. So, what exactly is a transformed cell? A transformed cell is one which contains a sort of a recombinant DNA. Now, how do you know that a cell has been transformed? If you want to exactly know how a cell has been transformed, you need to look out for where does it have an ampicillin gene. And if it has got a sort of an ampicillin gene, then you call the ampicillin gene as a sort of a selective marker. So, if we had your gene of interest and it is attached to the sort of an ampicillin resistant gene, in a PBR 322, if you have the foreign gene of interest and it is located near to the sort of an ampicillin gene, these two together, if it is introduced into a sort of a bacterial cell, that is, you have a plasmid and this is called a sort of a vector. You have your gene of interest as well as you also have your AMPR, and the AMPR along with this is introduced into a bacterial cell. It is introduced into a sort of bacterial cell. So, when it is introduced into a bacterial cell, because it has got ampicillin resistance, there is growth of the recombinant cells That is, there is increase in cell number and also cell size in a medium which is ampicillin resistant. So, in an ampicillin resistant medium, you think you have a nutrient medium and this is the sort of a medium which is also ampicillin resistant. So, you have your recombinant cells and the recombinant cells show growth because they have the gene for AMPR. They are ampicillin resistant. So, growth of the sort of an ampicillin resistant cells occur down here, while if you take the sort of a non-transferments, the non-transferments, because they do not have the recombinant DNA, they will not grow in the sort of a medium. So, what we are trying to derive is, for selection, AMP is termed as a sort of a very important, it is termed as a selective marker. So, the gist of whatever I am trying to tell you is, what is the first genetic marker when you do selection of transformed cells? The first selective marker is an AMPR gene. Next, we have limitations. There are limitations in the selection and the screening of the recombinants. There are limitations whenever we do screening and whenever we do selection of recombinants primarily due to inactivation of the antibiotics. Think we have a gene of interest. Now, supposing your gene of interest or your recombinant gene gets into a sort of a PBR 322, which has an ORI, and it has got a tetracycline resistant marker, and also it has got an ampicillin resistant marker. Now, if your recombinant DNA of interest goes over here, it can block this, or if it comes over here, it can block this. There can be situation number A and also situation number B. There can be two situations. So, every time whenever you are trying to screen a gene of interest, you need to test the gene of interest on a sort of a nutrient medium, which is either having AMPR or which is having TTR, which is tetracycline resistant or ampicillin resistant. So, every time a gene which has to be tested will have to be cultured into this AMPR culture, as well as it is also got to be cultured in a TET culture. Thereby, this process is terribly cumbersome. It is long drawn. It may not give immediate results. And at the same time, it is very important that your recombinant DNA requires to go through simultaneous plating. That is, you should always be checking in A and B, A or B, again primarily with relation to A plus B minus and A minus B plus. In different permutation and combinations, you should be selecting your sort of a gene. So, here whenever we plate here, we understand that whenever we are doing this sort of a plating, this will indicate whether the antibiotic gene AMP or TTR, AMPR or TTR is activated or it is inactivated. Inactivated means, supposing we have a recombinant gene and it attaches a TTR site. TTR, tetracycline resistant gene, can either be inactivated or it can be activated. So, it is a big sort of a process. Continuing on selection of transformed cells. Now, exactly where does the foreign DNA ligate, or where does the foreign DNA or the foreign gene ligate or attack? So, the foreign gene, whenever you have a foreign gene of interest, it can ligate. When you say ligate, it means it can join. it can join at a restriction site. It can join at a restriction site. Like what could be a typical restriction site? A restriction site could be one like if which is cleaved by BAM H2, which is cleaved by means of BAM H2. It has got a restriction site and it ligates at the restriction site in one of the two antibiotic resistant genes. If you understand PBR 322, in PBR 322 we have got a TETR as well as an AMPR. These are the two important selection markers. So, in these selection markers sometimes we have got restriction enzymes like BAM H2 which can cleave the sort of which can cleave the antibiotic resistant gene. So, when it cleaves the antibiotic resistant gene, this is the sort of a restriction space at which your foreign gene of interest comes and gets loaded. So, it is ligated at the site of BAM H2, especially if you take at the gene which is called TET gene, tetracycline resistant gene, in the sort of a vector, which is termed as PBR 322. So, while you are doing the selection of transformed cells, we understand the vector is PBR 322, the gene is TET, the site is BAM H2 and it is one of the sort of an antibiotic resistant genes, and we are looking in terms of what exactly is the sort of a process. Now, so once we have a recombinant DNA and the recombinant DNA is loaded into a sort of a vector, there can be two types of vector molecules which are getting formed. So, next we are looking in terms of vector molecules. So, vector molecules could be of two types. When you take the vector molecules could be of two types. One is sometimes the vector is unaltered. It is an unaltered vector. That means the foreign gene has not been inserted. It has not been ligated. The foreign gene does not enter into a sort of vector. So, it is not inserted and ligated. So, it has got only the sort of vector sequences. The vector DNA sequences are only present. Then we have a second situation wherein you have the vector and the vector molecule has the recombinant DNA. That is the gene of interest. The vector molecule has got your recombinant DNA or your DNA insert. It has a DNA insert. Now, when it has got the DNA insert, it is termed as a sort of a chiameric gene, or it is termed as a chiameric vector. It is termed as a sort of a chiameric vector, and or it is also called as a sort of a recombinant vector. So, a recombinant or a chiameric vector has got the DNA insert, and sometimes it can take in the sort of a DNA insert, or else in some case it remains unaltered. Now, both these sort of vectors are mixed and present in any particular sort of a cell. So, if we want genetic transformation, if we are looking in terms of genetic transformation of cells, wherein you have a gene clone, that is your recombinant DNA or your recombinant DNA can be multiplied and also expressed, because a DNA will have to make a sort of an RNA and an RNA will have to make a protein, and the protein makes the enzymes. So, it need to be multiplied, it need to be expressed, it is called a gene clone, and this entire process is termed as genetic transformation. So, prior to genetic transformation, how are the sort of steps? you have a foreign gene, it's ligated with into a restriction site into one of the antibiotic resistant genes. The vector is PBR322. We are looking in terms of vector molecules in terms of two situations, unaltered as well as altered. Altered means it has got a DNA insert, it has got a sort of chimeric gene or a recombinant vector, and we are now turning into what exactly happens during genetic transformation of cells. So, we have a normal unaltered vector. Now, if there is a normal unaltered vector, then the cells which are derived from that are termed as non-transformants. So, what are non-transformant cells? Non-transformant cells are those cells in which the vector remains unaltered, and because it is non-altered, these particular vector, these in the non-transformed cells, if we have a PBR322, imagine we have a PBR322 and the PBR322 is a vector, it is a plasmid vector. It has got an EMPR and it has also got a TETR. It has got an ampicillin and also a tetracycline resistant gene or a selective marker. Because it is unaltered, these cells, the vector can primarily grow in both the mediums which is rich in EMPR and TETR. So, this is a very important sort of point for you to understand. When you take a PBR vector, it has got a TETR and it has also got an EMPR. Because it has got tetracycline resistant and because it has got ampicillin resistant selective marker, this vector can grow in a medium which has got ampicillin resistance, and it can also show growth in tetracycline resistance because it is resistant to these particular two things. So, they can grow in both the mediums, in mediums which are ampicillin resistant as well as in tetracycline resistant. Your PBR322 can very easily grow. Now, this is situation number 1. Now, this is about primarily what happens in the case of non-transformed cells. How is the sort of a growth in EMPR, especially with relation to PBR322. While looking at the selection of transformed cells, if ever we are to study what is the sort of a recombinant vector. Now, if we have a recombinant vector, a recombinant vector is one which has got the foreign gene. It has a foreign gene. Now, if the recombinant vector is inserted into the site of TTR, if it is inserted into the site of TTR, due to the insertion, the TT resistance is lost. So, it becomes TT minus because it interferes with tetracycline resistance, and thereby this particular sort of a recombinant gene cannot grow in a medium which has got TTR. It cannot grow in a medium which has got TTR, but still the recombinant gene can be selected. It can still be selected from non-transformants. It can be selected from the non-transformants because it can always grow in a medium which has got ampicillin resistant. So, it is positive for ampicillin resistant and it is negative for tetracycline resistance. So, the transformants, if you think where the foreign gene is present, it is termed as a sort of a transformants. So, these transformants can show growth. The transformants can show growth, especially in a medium which is rich in EMPR. Now, if the transformants are placed in TTR, if the transferments are placed in TTR, they show no growth. They show no growth. Therefore, we can conclude that one antibiotic gene, if you recollect in the beginning of the lectures I was trying to tell you, one ampicillin resistant gene, that is AMPR, one antibiotic gene, helps in the selection of the transferments. Because there is AMPR is a selective marker, that's one of the reasons why you see an antibiotic gene like AMPR helps in selection of the transferments. Whereas the another antibiotic gene, which is called TTR, gets inactivated. It gets inactivated due to insertion of the foreign gene, and because of this insertion, because it gets inactivated, it again helps in the selection of the recombinants. So, this is a very important sort of a principle, like one antibiotic gene, that is AMPR, helps in the selection of transferments, and another antibiotic gene, that is TTR gene, is inactivated due to the insertion of the foreign gene and also helps in the selection of the recombinants. Now, so till now we were dealing primarily with relation to selection of transferments. After the selection of the, in one of the techniques in the selection of the transferments, there is another technique which is termed as colony hybridization. Which is termed as colony hybridization. Before we go into the colony hybridization, it is very important that you understand two important terms. One is termed as insertional inactivation. Insertional inactivation means when a foreign gene is added, the DNA gets depressed or it gets off. So, because of the insertion, there is inactivation of the particular sort of a gene. Now, colony hybridization is another sort of a technique wherein primarily what happens is you take a sort of a probe DNA. Now, what exactly is a sort of a probe DNA? A probe DNA is a small piece of DNA which can go and exactly align itself near to the sort of a recombinant gene. It is termed as a sort of a probe DNA. So, you have a probe DNA, and this probe DNA can combine or hybridize with the DNA of a bacterial cell, and it can hybridize, it can hybridize with the bacterial cell colonies on a nitrocellulose filter, So, the probe DNA the principle in colony hybridization is we have a probe DNA. The probe DNA is complementary, to at least one part of the desired DNA. That is, the probe will go and align itself with relation to your sort of a recombinant DNA. So, in this particular technique, primarily if we send a probe, the probe will go and align itself with relation to sort of a bacterial DNA. You have a probe, the probe and the bacterial DNA will align itself. It will align itself on a sort of a film which is termed as a sort of a nitrocellulose filter. So, thereby we can pick up the recombinant bacterial colonies. We can exactly know how to pick up this. So, if this is a sort of a nitrocellulose filter, these are the sort of a bacterial colonies which can be picked up. Thereby, we understand in the case of colony hybridization, there is no need of a genetic marker. There is no need of a genetic marker. And again, this technique of colony hybridization has been studied by Grunstein and Hogness in 1975. It is termed as an in situ, that is in the living state, hybridization assay. It is termed as an in situ hybridization assay. So, thereby here primarily we understand that if we want to screen recombinants, that is, you want to exactly detect how many recombinants are there. You want to screen recombinants which have a sort of a desired gene, with the desired gene from a sort of a gene library, from a collection of gene library. A gene library is a collection of clones. It's a collection of gene clones. Whenever we have lot of recombinant molecules, imagine a room with lot of petri dishes, and in all the petri dishes you got a gene of interest which is amplified, which is put over there, and you want to select which is the most important recombinant gene. Then from the gene library you can do screening and you can select your desired gene. When you do screening and select your desired gene, you can practice in situ hybridization technique. So, in situ hybridization technique, you are only sending a small sort of a probe. The probe will anneal along with the sort of bacterial DNA colonies, and it is seen on a sort of a nitrocellulose filter, and there is no genetic marker. So, it is when you say colony hybridization, it is more of a sort of a screening technique. Initially, it is a screening technique and than a sort of a selection method. So, though it is also termed as a selection method, the term selection method is secondary, whereas primarily it is a sort of a screening technique and later on it becomes a sort of a selection method, colony hybridization. So, here what happens is the probe will combine with the DNA, thereby you have a DNA combination on a nitrogen gel, or else you can have a DNA RNA print. It can be a DNA or a DNA RNA combine. So, this will be the sort of a print of a hybrid on the nitrocellulose. So, these prints on the nitrocellulose are seen in the form of dark spots by an X-ray film. They are seen as dark spots. So, these dark spots can easily be picked up. They can be picked up. So, each picked up spot is a sort of a DNA RNA hybrid or a DNA hybrid, and it can be cultured for further use. So, thereby here we understand that in colony hybridization is a faster technique. It is a faster screening technique, and the selection comes down here. When you pick up the sort of a cultures, after you see in the form of dark spots, this process is called selection. So, in colony hybridization, which is a screening technique discovered by Grunstein and Hogness, in situ hybridization assay, it is a sort of an assay technique. It is a sort of an assay. When you say assay, it primarily means you are detecting how much. Quantitatively, you are looking in terms of how much of the sort of by sending a sort of probe, how is the sort of an annealing on a sort of a nitrocellulose filter, how does the DNA and the DNA join, or how does the DNA and the RNA join. So, we first pick up in terms of bacterial cell colonies. Later on, we look in terms of DNA-DNA combinations or DNA-RNA combinations. And later on, we practice a sort of a selection method. And after picking the cells, we need to culture them. So, these are the different sort of a techniques how you do the sort of a selection of transformed cells. By selection of transformed cells, first method is primarily by using of a sort of an antibiotic resistant, that is AMPR or TETR. Then later on, the second technique is, because it's a very sort of a cumbersome method, you use a sort of faster method like a sort of a colony hybridization.